Dear students, today we are going to study a very important phenomenon of toxicology, that is the study of process of distribution of toxicants in a mineral body. Have you ever thought that why does mercury mainly affect the nervous system? Why pesticides often accumulate in fat tissues? The answer lies in one of the most important concepts of toxicology, the distribution of toxicants. After entering the body, every toxic substance follows its own pathway, influenced by blood flow, chemical properties, and organ affinity. Understanding this journey helps us predict toxicity, organ damage, and even survival. Dear students, as you know, toxicokinetics is the study of 5 processes including absorption, distribution, storage, biotransformation, and elimination. In previous lecture, we have studied the process of absorption of toxicants. After absorption through bloodstream or lymphatic system, xenobiotics enter other body organs. Lymphatic system comprises of lymph capillaries, lymph nodes, aggregations of lymphoid tissue, tonsils, spleen, and thymus, circulating lymphocytes, one of the 5 different types of white blood cells, while cardiovascular system comprises of heart, arterial vessels, venous vessels, capillaries, blood, circulating medium, blood cells, and in a yellowish, non-cellular fluid called blood plasma. Distribution of toxicants depends on dose, duration of exposure, chemical characteristics of the toxicant, presence of lymphatic or blood vascular components, precise location of entry of toxicants into the body. If we talk about distribution of dietary toxicants through cardiovascular system, toxicants that enter the animal body through oral route move to the submucosa of gut, enter the blood vessels there and through portal vein to the liver that act primary site for biotransformation or metabolic detoxification. From liver it moves towards heart, from where through pulmonary circulation it enter the lungs and through systemic circulation moves towards other body organs. Distribution through lymphatic system. Toxicants enter the lymph containing lymphatics which are associated with the digestive system, then enter the afferent, prenodal, lymph vessels, moves towards lymph nodes. Then through efferent, postnodal, lymph vessel go to the right and left YMPH trunks. Both these lymph trunks empty into the venous blood and through right and left jugular veins they enter to the heart and then systemic circulation. Next we will study distribution of respiratory toxicants through cardiovascular system. Toxicants that enter to the animal body through respiratory system transported from the lungs to the heart through pulmonary circulation. Then from heart to the systemic circulatory pathway, then distributed to the tissues of the body. Other inhaled toxicants, such as particulates, move into the spaces between cells, where they are transported at a slow pace along with the lymph in lymphatic vessels. Distribution of cutaneous toxicants through cardiovascular system. Toxicants absorbed through the skin enters the peripheral blood. Keep in mind that additional processes that affect toxicokinetics, such as storage, biotransformation, and elimination, may lower the concentration of the toxicant. Factors affecting distribution, physical and chemical properties of the toxicant. Concentration of the toxicant in the blood and in the tissues, i.e., concentration gradient. Volume of blood flowing through a specific tissue. Tissue specificity or preference of the toxicant. Presence of special barriers to slow down toxicant entrance. Properties of the toxicant. The same physical and chemical characteristics that determine a toxicant's initial absorptive behavior also determine its likelihood of being distributed to tissues in the organism. Distribution to tissues requires the toxicant to once more interact at the level of the cell. This time, Instead of interacting with the cells that form the absorptive barrier, its interaction is with the cells that form the tissues of the target organ. Molecular weight and polarity are again of concern, with the smaller, non-polar toxicants gaining preferential entrance. Concentration gradient. The concentration gradient between the amount of the toxicant in the blood as compared to the tissues will depend on a number of features. Following absorption, a toxicant will be diluted by the fluid volume present in the organism. This fluid is found in 3 separate sites: blood plasma, which accounts for about half of the blood volume depending on the individual sex and age. Blood volumes in humans range from 4 to 6 liter and typically will account for 7 to 9% of the total body weight. Interstitial fluid volume, the fluid between cells, which makes up about 13% of the total body weight. Intracellular fluid, the fluid inside cells, which accounts for about 40% of the total body weight. Dilution and the resulting concentration of a toxicant in any of these 3 sites is an important consideration, since the passive diffusion of the toxicant into or out of these sites will be determined by the toxicant's concentration gradient. A xenobiotic after entering the blood distributes throughout the body. Volume of distribution, VD. The apparent volume of distribution, VD, is used to define the volume of body fluids in which a toxicant is distributed. It can be calculated by dividing the dose of toxicant by its plasma concentration. Low volume distribution indicates the higher amount of toxicant in the plasma, while high value of volume distribution indicates low amount of toxicant in the plasma in addition to interstitial intracellular fluids. However, the apparent VD does not indicate in which of the 3 fluid volume sites a toxicant is distributed. The validity of the apparent VD can be further compromised by toxicants that undergo rapid storage, biotransformation, or elimination. Additionally, some toxicants bind to plasma proteins, example albumin, circulating in the blood. Binding to plasma proteins removes the toxicant from added interaction with cells. This removal effectively reduces the concentration of the toxicant in the plasma or VD, since only free, non-protein-bound toxicants will interact with cells. Times distribution depends on blood flow, rate of diffusion out of capillary bed into cells, affinity. Blood flow. The volume of blood flowing through specific tissues or organs in the human body is an important factor affecting distribution. 2 parameters affect the accumulation of a toxicant in an organ. Firstly, the volume of blood flowing through the organ. Secondly, the size or mass of the organ. A combination of these two, the blood flow-mass ratio, permits the comparison of toxicant accumulations in different organs. The percentage of the total cardiac output received by each of the body organs is also important, since organs that receive larger blood volumes can potentially accumulate more of a given toxicant. Cardiac output equals the volume of blood pumped per heartbeat, stroke volume, times the heart rate, beats per minute. During exercise, cardiac output increases as a result of a combination of a larger stroke volume and an increase in heart rate. Blood flow rate and distribution. Body regions that receive a large percentage of the total cardiac output or have high blood flow mass ratios, i.e., ml/kg, include the liver, kidneys, heart muscle, and brain. Blood flow to the brain is constant even during exercise. Approximately half of the total cardiac output will be sent to the liver, that is 27.8%, and for kidneys it comprises of 23.3%. This is not surprising, as these are the major organs involved in the elimination of toxicants or their metabolites. Skeletal muscle and skin have intermediate blood flow mass ratios. However, During strenuous exercise, blood flow to skeletal muscles may increase from less than 750 milliliter per minute to more than 10,000 milliliter per minute. This increase in blood flow may subject skeletal muscles and other organs to greater toxicant exposure and accumulation. Bone and adipose tissues have relatively low blood flow mass ratios. This is important to remember, since these areas also serve as primary storage sites for many toxicants, especially those that are fat-soluble as well as those that readily associate or complex with minerals commonly found in bone. Affinity of toxicants for specific tissues. Some tissues are attractive to specific toxicants and, in spite of the rather low flow of blood to these tissues, they will preferentially accumulate a given toxicant to disproportionately high concentrations. A good example is adipose tissue, such as subcutaneous fat, which is poorly perfused by blood but is an attractive tissue for lipid or fat-soluble toxicants. Once deposited in these storage tissues, toxicants may remain for long periods of time due to their solubility in the tissue and the relatively low blood flow. Structural barriers to toxicant entrance. During distribution, the passage of toxicants from capillaries into tissues or organs in the body is not uniform. Some organs have specialized barriers that make it difficult for toxicants to diffuse into their cells. Blood-brain barrier, placental barrier. Blood-brain barrier only serves to slow down the rates at which toxicants cross into brain tissue. This barrier is formed by specialized cells. Glial cells, astrocytes, star cells, have thread-like branches and end feet. Tight junctions between adjacent endothelial cells limit the passage of water-soluble molecules. Astrocytes attach through end feet to the outer surface of the endothelial cells that line the capillaries, forming a barrier that separates the endothelium of the capillary from the neurons of the brain. Lipids in the plasma membrane of astrocytic end feet impede the diffusion of water-soluble toxicants into the brain. Lipids in the plasma membrane of astrocytic end feet impede the diffusion of water-soluble toxicants into the brain. Placental barrier. This barrier is formed from cell layers between the maternal and fetal circulatory vessels in the placenta. Lipids present in the plasma membranes of these additional cells serve to limit the diffusion of water-soluble toxicants. As with a blood-brain barrier, the placental barrier only slows down the diffusion of toxicants from maternal circulation into fetal circulation, reducing exposure of developing fetal tissues to toxicants. Thanks for watching. Like and share this video. Subscribe this channel to get more informative videos.